Obesity and inflammation, that being obese, special central obesity, is causing chronic inflammation, it's well known. However, we didn't know exactly how this excessive accumulation of fat was responsible for this immune activation, macrophages activation, causing the release of pro-inflammatory cytokines and systemic inflammation that here they call meta-inflammation. Now, this very interesting paper just published on PNAS by the group of Sweden Burgdorf is shedding some light on the mechanism. It's very, very interesting paper. So just to summarize, if you are in a hurry, what they are finding is that soluble mannose receptor, called SMR, plays a direct and functional role in activating macrophages. So basically, SMR binds the CD45 on macrophages and inhibits phosphatase activity, causing an activation of AKT and FKB that reprograms the macrophages to become pro-inflammatory, both in vitro and in vivo. They also show in this paper that serum concentrations of soluble MR are increased in both human and mice that are obese and directly correlate with body weight. Most importantly, this soluble MR is causing an increase in pro-inflammatory cytokines and activation of macrophages, both in the white adipose tissue and in liver, and promotes systemic insulin resistance and therefore aging. So showing that inflammation and insulin resistance, they are strictly related and in part caused by this obesity-related increase in soluble MR. Okay, so this is the summary of this paper. If you want to understand a bit more on the beauty and the experiments in this paper, stay with me. So let me show you here on figure 1 the effects. So these are macrophages, wild-type macrophages, or macrophages with the knockdown of the mannose receptor. And as you can see, those that when you knock down the mannose receptor, you have a much lower production of TNF-alpha, interleukin-6, and interleukin-1 beta in response to LPS stimulation. This is again murine macrophages, but even in human macrophages, if you expose human macrophages with recombinant MR mannose receptor, you have a huge increase in TNF-alpha production. Okay, so this is showing the effect of these soluble MR. In figure 2, they also show that this soluble MR is causing a wide increase in gene expression of multiple pro-inflammatory genes. This has been done by RNA sequencing analysis in macrophage treated with MR. In figure, let me see, figure 3, they show that the mannose receptor inhibits the CD4 activity on macrophages with consequences in the secretion of TNF-alpha, interleukin-6, interleukin-1 beta in response to LPS. And in this figure, that is figure, let me see, figure 4, they show that this mannose receptor is causing a change in the macrophages with an activation of the AKT and FKB pathways. And FKB is the major pro-inflammatory pathway within our cells. Here are all the experiments, beautiful experiments. So it's a very well done study with a lot of data. And this is very important. So here they show that in mice, as you can see here, a high-fat diet is a model of obesity. So if you feed animals a high-fat diet, that is hypercaloric, these animals become obese. And as you can see here, in these obese mice, there is an increase in soluble MR, but not in those where MR is knocked down. And there is a nice linear correlation between soluble MR and body weight and body fat. But even in humans, obese individuals, they have higher level of soluble MR, so circulating MR in their serum. And again, you know, there is a nice relationship in obese individual between soluble MR in serum and BMI. Okay, so showing that when you are obese, you have an increase in circulating soluble MR. And this circulating soluble MR is not just a marker of obesity, but as you can see here in this study, when they knock down the mannose receptor, especially in animals that have been fed a high-fat diet, this is causing an improvement in HOMA, that is a marker of insulin sensitivity. As you can see here, the animals, the wild-type animals on a high-fat diet, they have liver steatosis, but those where the mannose receptor is knocked down, they don't have the steatosis in response to a high-fat diet. Huge reduction in steatosis and liver triglycerides. And also there is an improvement in glucose tolerance and area under the curve for glucose, as you can see here. And finally, here we see in this figure 7 that if you take normal mice, wild-type mice, on a normal diet, and you inject twice a week mannose receptor in these animals, there is a pro-inflammatory reaction. So there is an increase in TNF-alpha, interleukin-6, interleukin-1 beta, MCP-1. There is an increase in the infiltration in the adipose tissue of macrophages that are producing more interleukin-6 and interleukin-1 beta and MCP-1. And by the way, we have shown that in humans, obese in humans, interleukin-6 produced by visceral fat is increasing systemic C-reactive protein. So beautiful, beautiful data. Just to summarize, okay? So this mannose receptor is a member of the C-type lectin family. And this mannose receptor has an extracellular region that can be shed by metalloproteinases and released as a soluble protein that, as you have seen, is the quantity of these soluble MR are higher in obese individuals. So people who gain weight, they have, or mice and humans who have gained weight, they have higher soluble MR. And these serum soluble MR levels, by the way, in other studies have been shown to increase in a variety of inflammatory diseases, and the concentrations are directly correlated with the severity of the disease and mortality for those inflammatory diseases. Now, in this study, this group have shown that this soluble MR is not just a marker, but plays a direct functional role in macrophage activation, especially in adipose tissue and liver. And these macrophage activations is causing, this SMR is causing a pro-inflammatory phenotype with secretion of pro-inflammatory cytokines, TNF-alpha, interleukin-6, interleukin-1 beta, that we know are causing insulin resistance. And the mechanism, as we said, is through inactivation of CD44, so CD45. So basically, the soluble MR is causing an inhibition of CD45 and an increase in AKT and FKB signaling the macrophages. And in vivo experiments, soluble MR are basically causing an acute increase in serum pro-inflammatory cytokines, and they induce both tissue macrophage activation and systemic insulin resistance. Okay, so beautiful, beautiful data. And the bottom line is that don't gain weight, don't accumulate excess body fat, because this is causing systemic inflammation, and systemic inflammation is bad because it is responsible for insulin resistance, as we've seen here, for liver steatosis and fibrosis, and so basically fatty liver disease, and also chronic inflammation. Systemic chronic inflammation is one of the main factors responsible for cardiovascular disease, dementia, many inflammatory diseases, many types of cancers, and aging itself. You know, if you have this chronic inflammation, you're going to age faster. So thank you for listening, as always.